Yeah, thank you. Quick question, who has already heard about open-endedness? Can you raise your hands? That's unsurprisingly larger number of people than usually for my colleague. This is like a very vague or weird idea, but here there is actually a track about open-endedness. So there are many ways of defining open-ended systems. The usual definition I have in my head is that an open-ended process, it's something that creates things, and these things are either always novel, so it can constantly generate new things, or it's able to constantly improve its function, whatever function means. And some people also define it as processes that do not have a certain target goal to move towards. These types of things exist. Think about biological evolution. There is no endpoint to evolution, or I think most people don't believe there is like a supreme organism to grow to, but this is like a process that constantly generates new organisms. These organisms interact with each other, they create niches. This induces like other types of organisms. So this is the prototypical open-ended process. But this also occurs in man-made systems. The scientific process is hopefully open-ended. Creativity, innovation, art, literature, and so on. These are things that we actually want to believe are open-ended, and we have a lot of workshops or sessions or things of improving creativity or this open-endedness or innovation or whatever. The way how we see this work, or our work specifically, so open-ended systems, this is something that is very much part of artificial life. It's obviously also studied purely from the biological side in evolution. And our ideas is like, how can we relate this to synthetic biology of making new biological functions or new organisms? Now, open-endedness, this could be like a thing that you would think like, it's something I know what it is, I would recognize it if I see it, but of course, people have spent lots of sessions of like clearly outlining this. It was actually discussed here, well, not physically here in Copenhagen, but on a previous session of the Artificial Life Conference in Tokyo. These are the Tokyo categories which we adapted for our viewpoints of biology or synthetic biology. So if you have an open-ended process, then you would expect unlimited generation of four things. Novelty, so new entities. For example, if you have this enzyme or certain enzymes, this is a P for 50, an enzyme that occurs in many types of organisms, from bacteria to plants to in our liver. These are very promiscuous. They can oxidize certain metabolites, and they can actually evolve very easily to process other types of substrates. So this is a form of unlimited novelty. This is about the entities. The process itself, if it's starting to become open-ended, we'd also expect that this process changes so that it's easier to change. And this is called the evolution of evolvability. If you think about this, as many people do, as like these fitness landscape, then what you would see is that this landscape becomes smoother, or you are moving to regions in which they are smoother and easier to optimize. Or if you have different models like these phenotype networks, means that they're just better connected, and then it's easier to explore new exciting phenotypes. Open-endedness also implies some form of macroevolution that is possible. So you would see major transitions. Think, for example, as like moving from prokaryotes to eukaryotes, or involving multicellularity. The latter is an interesting thing also in synthetic biology, because there are a couple of different papers with different approaches in which they do have evolved in a couple of hundred generations, a unicellular organism into a multicellular one, making some kind of woody substrates from yeasts. And the last one is semantic evolution, or this term mainly from language, but if you think of biology, it means like functional chains. My favorite example is CRISPR-Cas. This is a defense system of bacteria against viruses, against phages, but we have like adapted it, biotechnologists have adapted this as a way of genetic engineering. A more recent definition or a more recent way of thinking about it, they see an open-ended system as it produces a sequence of entities that for a certain observer is both novel and learnable. And these are the two properties, two kind of contrasting properties that it should satisfy. For example, if you think like production of art, this is open-ended. You have these new paintings or new painting styles that are produced. And if you would think, okay, this sequence is open-ended, what does it mean in practice? Well, it means that if you think you are at a certain time point, things that are in the future, in the very far future, paintings from the future, they are novel in the sense that they are very hard to predict. Or if you use some kind of complexity, they are very surprising. So this is novelty, but you also need learnability. And this means that if you see a new painting just after now, then the more of the history of your sequence you have, the more of art history you know, the less surprising it should be. This is what it means to be learnable. And this definition, to me, this makes it very clear to think about, okay, why is something open-ended? Why is it not open-ended? If I start with this art exhibition with these paintings that I generated, these are all random pictures, random noise. I'm very certain these are all novel to you. You have never seen them before, but this process is all very novel, but it's not learnable. You will not be able to predict the next image, unless, and this is a philosophical discussion, as you know, like the model is just rand n, and it's generated depending on your definition of what randomness is, but this is clearly not something open-ended. This is a different example. Here I generated images, and some of you will likely recognize it, using one-dimensional cellular automata. And this, in the beginning, might be a little bit novel, up until you see the pattern and you say, oh no, it's just using this very simple code to always generate images. So at this point, they don't become novel anymore because you can perfectly predict it. And I like this example because it also illustrates that the observer is quite important. So somebody who knows what cellular automata is, and if you already did this before, then it's definitely not novel for you, or you will immediately recognize this and you can produce similar images as well. But if this is something totally new to you, then you will likely need to see more images or be able to see the pattern, and before you can start making these. So observer, sequences, novelties, and learnable, these are the key concepts. Now, if you follow the logic of these systems, if you make them a little bit more precise using things like algorithmic information theory, then you can actually deduce some very nice properties of this. If you have a system, these entities, they're very often consisting of basic elementary building blocks, Lego bricks, that you can combine to certain entities. What you will see in open-ended systems, there will be, or very often, it's not a necessary requirement, you see emergence that you can combine your building blocks, your atoms, into higher order building blocks, like in molecules, which you can again combine into higher order structures. So this is a way of like generating open-endedness. And if you have this certain tension between the sequence of these entities, that they should be novel on the long run, but very predictable on the short run. If you make this precise, then this tells us something about the distribution of these building blocks that they have to satisfy. And if you do the math, done in this article, then a zip law of the building block distribution emerges. So it knows that if you have an open-ended system that uses certain building blocks to combine new things, then the distribution of these building blocks of the frequency has to follow this law. And this law is most familiar or is most well described using word ratios. Language is open-ended. You can never write all the texts. But this also occurs in natural or in man-made systems. For example, protein domains. This also follows this zip law, or if you have electrical components, Lego bricks, it's shown also by ingredients used and so on. And yeah, if you look at this, you can show that certain forms of information, they get lost over the long run, like the statistical information, Shannon information, this is lost, but algorithmic information is retained. So you can play with this. Now, making systems, there are like two general ways of making systems or having these processes. You can either design something or you can evolve something. And like a great way of thinking about this is if you have things, and typically they can either be machines, things that we built ourselves, or they can be organisms, things that have been evolved. And this is, of course, not a perfect distinction between these things, but I found very interesting to have this in the lab. So when we have a machine, think of a toaster or a laptop or a chair or whatever, then this is made by people, and this is made to be easily to manufacture, predictably to use, to repair, and so on. So this will behave quite linear and predictable. This is usually assembled of parts, and there will be a fixed number of parts that are used. Like not when you build IKEA cupboard, and you have some spare parts, but usually it's fixed, and it's designed from a blueprint. So some intelligent agent has thought about how to make this. And very often, machines are quite general, so they can do many things. And they do have some kind of software, or many machines have software, which is very distinct and somebody can program. Now, if you compare this with an organism, with like a plant or some kind of animal, this is very unpredictable, this is very non-linear. And it doesn't have a fixed number of parts in general, like it's composed of cells and it can grow more cells if it needs so. So there is a lot of flexibility. These are evolved. And instead of being modular, modular to some extent, they're hierarchical self-similar. So you do have this emergence of different complexities. And there is far less hardware-software duality. There is embodied intelligence. So we do have this high school notion of, okay, DNA is the carrier of information. This is in the cell. Or we have this idea, okay, the brain, that's our computer. That's where the software is. But if you look a little bit deeper, this is also something that Susan has mentioned. This is not the entire picture, because everything that happens in biology also is used to some extent for computing. Like the membranes in a cell, they are part of the information processing, or limbs and so on. This is also part of like our processing. And this duality, this is an interesting thing, because I think one of the successes of synthetic biology is just by looking at a cell or a bacteria, not saying, oh, this is a wet clump of biology we cannot understand, but this is about thinking about a machine with like individual parts. It has genes I can understand, I can modify, I can predict, and I can design it. But on the other hand, a lot of our technology, like yesterday was here a session about evolution of things. We are more and more thinking about hardware or machines are these things that can be evolved organically. Or think like our current iteration of artificial intelligence, neural networks, these are basically evolved or grown under the right conditions. And so machines or organisms or basically any entity, you can basically put it like on this square. You can say, okay, this is either something that is evolved or it's something that is designed. And whether it is like a clockwork, like a program, so it's programmed, or whether it's something capable of cognition, so more cybergenetically. And since both machines and organisms, they exist at different levels of organization, what holds for like one level does not necessarily hold for these other levels. For example, you have a cell, which is a very living, evolved thing, but you can combine cells into a machine that is relatively predictable. Or you can have little robots, which are very predictable and simple, but you can combine them into a swarm that is much more organically and acts more like an organism. And this is, for example, a nice example of making a machine out of cells. This is the xenobot example. So here they try to make reliable machines from cells, but they do use an evolutionary algorithm, or they do use ideas of evolution to design this. Now, evolution, this is indeed like the thing that is used a lot. There are many sessions here about evolution. You typically need three things to have a process that evolves, either in silico in your computer, or in the lab, or in real life. So you need a mapping between a genotype and a phenotype. This is quite important to have a distinction between the two. You need a way of creating variation in your genotype. Random, this does not depend on functionality or objective typically. And you need some kind of selection mechanism, either positive, you pick the best one, or you eliminate the weaklings. And if you have this process, then you have evolution, then you can use this to improve something. Now, when I wanted to do my PhD about evolutionary algorithms, then my supervisor said, like, don't do it. I tried, it doesn't work. There is, yeah, obviously it does work, but a lot of people, when they try evolutionary algorithms, they are a little bit disappointed in the sense that, like, you kind of know that everything around you is made by evolution. And very often when you try a naive implementation, then this does not work as well as you would initially hope. So maybe there is a little bit thing that you can do to improve this, or this is something that is not a complete picture. What you will see is that, yeah, this we call objective-based evolution and design. So based on how well it performs, or based on the fitness in nature. And for example, if you use this to design these phylogenetic networks or oscillators or something, then you're going to think about this as a fitness landscape, and you want to attain like high fitness. You want to find these big peaks. Now, what you often see is that if you start from an initially diverse population, which cover like part of this genotype space, then you will see initial improvements in the fitness or the activity, but after some while, this has diminishing returns because you're starting to zoom in on like a local optimum, and your diversity plummets, and improvement stops. And this is not really what we want. This is not really open-ended, and there are maybe these very interesting regions here, which are very hard to find on this way. And evolution or artificial evolution, and this is explored in a lot of open-ended in artificial life papers. This does have some well-known bottlenecks that this naive version maybe has a bit of problems with. One thing that is discussed in this paper, examples, also it's discussed in a paper is the reality gap problem. The idea that your artificial environment, it does not completely reflect the natural environment. You have this very cool paper, surprising creativity of digital evolution, of like this artificial evolution, usually of robots, and where it fails because it just exploits the simulator for your objective. Here they try to evolve robots that could move fast. So the thing that the simulator, like the evolutionary algorithm came up with is, okay, I'm just going to make a very long pole with like a mass, and then it falls over, and yeah, it has moved a little bit, so that's a good step. And that's initially a good idea, but that's not a very great working point for having a robot that can walk on a complex terrain. But this is the reality gap problem, but also just a different problem. This is called the stepping stone problem. This means that if you want to evolve something, if you want to attain a certain function, then the path to this might not be initially obvious, or it might only be obvious in retrospect if you look back. This is illustrated very nicely with the big reader. Maybe some people who were in the session before me at neural evolution is also discussed here. So big reader is basically a neural network to generate images with quite a bit of structure and symmetry. And you can generate very nice pictures of all kinds of everyday objects. For example, of this skull. But the problem is, if even though you know there are in this space, the skull is present, it's very difficult to actively look for this because the intermediate steps, they don't necessarily look like this. So if you just use a regular evolutionary algorithm, you just have an objective, like how much does it look at the skull, then you end up with just the very boring pictures. But if you use more modern variants like the quality diversity, or you use novelty, just look for very exciting or novel ones, then you will explore this space more systematically, and you will end up at some point at something that maybe resembles a skull, or you can use to make such a skull. And this is a little bit the point that is on the other hand of the spectrum. So either you have functionality or fitness, or you have novelty as a basis. How new it is. This is argued very nicely in this book that I think a lot of you don't know, My Greatness Cannot Be Planned. And they say if you just explore something for novelty, then you will be able to, or in theory, you should be able to cover the space of like all types of behavior that is possible. And as a byproduct, you will also get things that are quite performance. So if you use novelty, finding new ways to do it, then what you would see is that you will be able to keep improving in theory, and your diversity is not exhausted. And this is like a big question, like novelty, it seems like a very good idea for selection. And the question is, maybe nature also has some idea that it uses novelty. Novelty is harder to use in practice than fitness, because novelty requires a brain to some extent. You can only assess novelty if you have some memory of what you have seen before, and you have some kind of way of comparing them and just saying like, okay, this is different. It's also relative. It's also a bit more difficult to implement a novelty score compared to a fitness. This idea of using novelty or diversity, this is explored in many papers. These are some recent ones. There's some follow-up work that I think will be presented in the following days. POET, for example, is a way of using evolution for new robots and their controller. And the strategy here is that you're still going to optimize for your robots, but together you're also going to do co-evolution of environments. And you're going to vary your environment and you're going to try to match certain robots with certain environments. And this should be something that you hope has some kind of complexity explosion and that you are able to detect like very interesting robots that can move in certain environments. OmniEpic is something that is also uses this idea of novelty, but novelty it's hard to define mathematically. It's a very human notion. So within this evolutionary algorithm here, they use large language models just to say, okay, this your current algorithm is capable of doing that, and to say what would be the most novel or the most interesting thing to do. So you can use these things together to can have like a large language model as a way of performing the selection or as a mutation, and this is a very powerful combination. Now, back to biology, why would we be interested into this? Well, we want to do design new things. We want to design something that we haven't come up with yet, or that doesn't even exist yet. We want to be ahead of evolution. And this is one case that we are working on in our group. We are working a lot on artificial intelligence for phage therapy. Phages, they're these little viruses of bacteria, the good viruses. And they can be very effective when antibiotics does not work yet. For example, in the burn wards, very often people have severe infections, and these are antibiotically resistant bacteria. And what they're doing is they try to find in nature phages that work well specifically for that bacteria. But this biological interaction is so specific that a lot of synthetic biology is used to tailor these different phages. What they do have found, or the generally know, is that if you have a phage with like this capsule over there, contains like the DNA and all the genetic information, but these little legs that you see over there, these are things that it uses to adhere at the bacteria, and that it uses to inject its DNA or RNA into the host. And by swapping out these little legs, these tail fibers, you can change the specificity. But this is a thing that is active research of like finding very good, or maybe even designing tail fibers towards specific strains of bacteria that you want to kill. And these are things that, there will always be new bacteria because they're evolving. So you need this kind of open-ended approach for this. So what we try to do is we took a look at like these very classical novelty search and quality diversity algorithms, and we were thinking like, how could we adapt it to synthetic biology? For example, if you have novelty search, this is in my head like the most pure one. Just take whatever is the most novel. This is something that you could use for discovery of new molecules, of new proteins, or functionality that you haven't seen yet. For example, if you want to find like new colored fluorescent proteins, just always pick on every generation that is a color that is different from what you have seen up to now. Or if you want to find new enzymes, then look for a specific one that's probably very hard to find. But if you have some assay, try to look for certain reactions that are novel and might be a very good starting point for things that might also be useful. There are variations that are a bit more performant or do use some objective, for example, minimum criterion, which has some minimal requirements of your objects that you need. In addition to being novel, this is something that you could also use. For example, if you're looking for new genetic circuits, look for ones that are interesting but do try to keep like the metabolic burden and how much energy they draw from the host to minimum. Or you have MOPI leads, which is the illumination algorithm. This is a very older one, also found it works very well. This is, in my opinion, one of the easiest to apply. in both in the computer and on the lab. So just create different niches, different of combinations of what your solution should require for, and take only the best one into this niche as a way of keeping diversity. For example, if you're trying to find some kind of new bacteria to produce some kind of metabolites, then you might create niches based on the number of knock-ins or knockouts or mutations or whatever. So some notion of complexity. And you might also compare them into different substrates or combinations of substrates and always take the best one of this combination. Or, and this is something that is actively also used in biotechnology research, you can use notion of co-evolution to speed things up. And this is something that we generally thought about and also looked into the literature. So you have these three pillars of evolution I talked about. So you have your variation mechanism, you have your selection mechanism, and you have your phenotypic realization, so your genotype-phenotype map. These are, of course, like your three pillars of evolution. But the reason that in nature that this works much faster and more effective than a very naive implementation of this is that these are not in isolation, but they influence each other and they are influenced by external factors. And I think as a bioscience engineer, the question is how can you create meaningful feedback loops between those? For example, as the bioengineer, or as a selector, we have the option of like turning up or down the amount of how much you mutate so that you can adapt this evolutionary process, or you can change the environment or something, or you can put in other entities for like all kinds of co-evolution that you can affect these processes. Or, and that's a very nice thing of synthetic biology, using genetic circuits, you can even allow the organism itself to modify these processes. But one thing that you can do with the genetic circuit is have like a very adaptive control of the mutation rate or like a kill switch so that it does its own selection. And this is like a general way also, like at a more higher level, the way how we think about design. This is something also that we could think about improving this. So this is the very classical build, test, learn, design cycle. This is most synthetic biology papers are based on this scheme, which, as Tom mentioned, has a very evolutionary feel to it. This is, of course, something that works great in practice, but this is very objective focused. So this is good for improving a certain function or making things better or tweaking it so that it works well, but it's very bad or does not generate new things. It should not surprise you. And there are other engineering paradigms that are a little bit more open-ended, that do take creativity more into account of this. And one of the concepts that we explore in our engineering as evolution paper is, for example, the CK theory, where you make a distinction between concepts and knowledge. And concepts are just properties of your objects that do not necessarily have a status that they're true or false, but just things that you could realize or concepts. And you have knowledge, things that you know, or information that you can get by literature or by doing experiments. And by interacting these two concepts, there is some more exploration that you can do, and you can get a more open-ended process. So I don't know, are there any questions or remarks? Or Then we have prepared an exercise. We have a didactic material. We have here our biodesign machine, and the idea for you is to design your own machine that will solve all the world problems or make you rich or whatever. A biodesign machine to solve like the big world problems, but one that is open-ended, so that can constantly improve or create new things. And yeah, you have this machine, and the idea is that alone or in two or in three, whatever you feel comfortable, that you pick like a certain topic or kind of application field. These are a couple of ones that we thought was interesting, like for example, making the phages or generating new crops or making novel foods or metabolites or new ecosystems, or you can go crazy and you think about what types of technologies or ideas or approaches could you use to have such a process. And here it suggests like it's a bit of a machine and desktop thing, but it does not necessarily have to be a machine. It can also be like an organization or like just a workflow or whatever. So we're just curious what you can come up with. And just to get you started, we also have some sheets where this is all up, so you don't have to remember this, but we have some biotechnology tools or things that you can use. We have some of the software, AI, or methods that might be useful, and hardware that you can combine. And yeah, you can think about this machine. So give it a fancy name. Think about what would be the input, what would be the output, what would be the hardware component, what would be the design or evolutionary component. So it's an open-ended exercise. In addition to this, if you were not quick enough to scan like the QR code on our website, so on the list of tutorials, there is a website with the overview of this tutorial. We'll also put our slides there, but there you will also find this link. Okay, let's take 15 minutes, and then the best ideas can like propose it.
Okay, everyone. So I hate breaking up this session, but because we've got 10 minutes left, I thought it'd be quite nice to use this as an opportunity to have a sharing part where we can see what sort of ideas people came up with. Is anyone willing to sort of like, I don't know, tell us a little bit about the machine that they thought about, or some of the things they thought about putting into it, or bringing them together or just get it to work? Okay, you be brave. You be really brave. I can see someone looking at me. You can be brave, go for it. Go on. The reason it's really useful is we really like to pull together each other. So like you can be crazy about it, like so this just came together like 1 minute ago. But we picked the phage board, and we kind of set up a co-evolutionary process between the population of bacteria. We kind of assume we have some bacterial generator. Yeah. Maybe our stomachs or something, but the bacteria are generating from new food sources, but then that bacterial population goes into where we put phages into phages reproducing in some of the bacteria out. So we have this like phage population, and we take some of those phages are going to be output a whole machine, and some of it just go back and do this. So the machine, here, it's going to constantly get new data.
Cool. Then, yeah, I think it's feasible, and also you can imagine, like, you see like where bacteria come from. We can create a diversity of bacteria in the DNA. So there's things we can do that make sometimes those sort of ideas quite feasible. So, okay, well, most importantly, what's naming your thing and the machine and whatever you process. We have like a.
Yeah, we don't. Maybe
Anyone else willing to sort of say a little bit about the approach? I heard you talking a lot. I think go for it.
Okay. It's maybe weird. Yes. So there's this creature that is monitoring some environment, collecting data to keep the environment safe. So there's a behavior required for the to go around the environment, and then there are biosensors and creature, and this biosensors can regulate genetic material that express traits, and then there's this embedded DNA microarray analysis that reads what's being expressed. And this is because what it's being expressed, of course, manifests behavior or changes in there, but it's also read by the DNA microarray. And this feeds to the gene gun that generates changes to the DNA. So the behavior that was, the behavior that led to certain data collection that led to expression can change the DNA itself. And in the future, the expression will be different. So this is feedback.
It looks like the organism can observe itself and then evolve in future generations based on its experience. Some sort of like evolutionary thing within the organism itself.
Adapt to the environment.
Yeah, that's really cool. Yeah. And yeah, some of those things like, so I always think of these things from the sort of biological, practical perspective. Some of those elements are possible in terms of like the modifications that they could be designed in, they'd be limited, not unlimited. Excellent. Anyone else? I'll see if you think back. There's a group here. Anyone willing to go in there? I think give us another way to look about.
So yeah, we decided to go with the light maker. And one thing thinking about it, how else we started is like if so essentially to try to tie the idea of like the metabolite that you want to some like critical process in itself. So let's say given the world of protein engineering with large language models, let's say we have that available to us. Like you have a metabolite you want, and I make like a key protein like DNA polymerase that's only active in the presence of that metabolite. And I design the active site to look like the metabolite essentially, right? So like when it's in there, it's going to work. And then if you can make that somehow like a continuous, so a better shape, like the metabolite like close to the design you want leads to like a better version of the like or DNA polymerase is on more, let's say, right? Then potentially you can tie like the construction of the metabolite to bits of the cell. And that way, like it will try to come up with better designs of that metabolite, but yeah. Or like better chemical processes to produce the metabolite, sorry.
Yeah, no, definitely. Yeah, I guess also like you can, so there are these technologies that allow us to kind of scramble parts of the genome as well. With metabolites, you often have lots of enzymes that are involved in carrying out conversions and things. You could imagine there being kind of almost enhanced variation being generated in that part, but you've got the selection part that works as well.
Yeah, something like that.
That's cool. That sounds great. Okay, I don't know what's the time. I've got like 2 seconds. I don't know. Yeah. We got time for one more, or are we gonna?
Someone has the ball more wants to do it.
I'm feeling great. No. But one of the things I was going to ask, we were talking about this beforehand, was we'd really like to kind of, I don't know, write up or like pull together some different perspectives that you have. So
we were hoping some of the stuff to get written down, or a lot of it was just general discussions between you. It would be great if you have an opportunity during kind of the rest of the conference just to make some notes in that Google Doc that we showed. So we can send an email around to anyone that's signed up with an email to include that. Like I sort of said at the start, we're really keen. If there are people here that want to get involved in putting together something, like a publication around some new ideas, more as a perspective to spark a community in some sense, or to help to get that established, then let us know. And I don't know, does anyone have ideas in terms of the things that they would like to do, or are you happy to discuss things offline?
Very, very quiet.
Do you have a story to know?
I don't think if we have it, I don't think tutorial yet, you can ask the organizer to settle that. That would be good. I think you do. Do we have one? I saw you. I think I want to take it to the next time we use this very well, maybe it's not that, but
If we don't have one, we will ask to make one.
yeah, definitely.
Is that one?
Okay.
Yeah, I couldn't find it when I looked, but we will have a look and we'll get them set up, and that maybe is a good way of collecting this information. But yeah, if you're interested in getting involved or helping, then let us know, either email us or something. I don't know if you